Hi everyone, and welcome to the first review video for the Biology or Living Environments regions. This is Unit 1, Topic 1. So just to quickly give an overview for all of these videos, the format I will go through is reviewing the content and then going over some review questions for the content we just did at the very bottom. All the review questions being done can be found online by just looking up Living Environments regions, and then you can get a copy of all the Regents exams for the past, 15 years freely online. So let's get right into it. The first subtopic for this topic is lab safety, and these are really basic rules. This is copied straight from your study guide, and if lab safety is on the test, you should be very happy. Lab safety is very simple and it's really common sense. Like if there are chemicals in the lab, you obviously shouldn't eat them. So I'll just read through some of these. If your teacher tells you something, you should always follow their directions. If you drop something or spill chemicals, always report the accident to the teachers and don't try to hide that you messed up and clean it by yourself. You should always wear appropriate clothing in the lab, like gloves, goggles, and whatever is recommended. And for anyone with long hair, you should tie your hair back to make sure that it doesn't fall into any chemicals or disrupt the lab in any way. This is an important one that people sometimes forget. When you're heating a test tube, you should always point it away from you or others because if anything starts bubbling or chemicals start spitting out, you don't want that to get on you. You should always wear goggles when heating or working with chemicals for the same reason, to protect your eyes. And finally, you should never stopper, which is another word for close, a test tube while it's being heated. As if gas is produced when it's being heated up, that's all going to get trapped under the stopper, and then when you finally open it, it's going to explode everywhere, which is no one wants that to happen in a lab. So always make sure you don't stop a stopper or a test tube while it's being heated. Okay, let's move on to the next thing, which is the scientific method. So for the scientific method, you want to know the steps of the scientific method to conduct your experiment. So the first step you want is to determine a problem. This can either be given to you in a lab, like a question you want to solve, or something you find from your everyday observations. For this entire lesson, we'll give an example of seeing how the amount of light affects the plant. So the second step you would do is do research on the topic. So this research on the topic could be looking up factors that affect your problem, looking up some important background, anything that might be significant for your experiment. Number 3 is you form a hypothesis. Your hypothesis basically an educated guess, which is your prediction of the outcome. That's not a question. This is often best phrased as an if ... then ... statement. So you identify if you change blank, which is your independent variable, then blank will change because blank. So for our example, we'd say if the amount of light increases, the height a plant grows will increase because more photosynthesis can occur and it'll generate more nutrients. And that because statement often comes from the research that you've done on the topic. So the next step number 4, is to design an experiment. You want to design your controlled experiment to find the relationship between the independent and dependent variable, and we'll look at how you design that experiment in a minute. Next is step number 5, where you collect and analyze your data. So you actually run your experiment, you take all the necessary measurements so you can collect your data and start to analyze it. And then your final step number 6, is to draw a conclusion. So your conclusion will restate the hypothesis. It will say whether your collected data supports or refutes the hypothesis. It will give an analysis of the validity of the experiment and can do a bunch of other things. So those are the steps of the scientific method. Now the next thing we want to look at is how to design a controlled experiment. So the first thing you want to know is the distinction between your independent and dependent variable. The independent variable is what is being tested. So it's really what you change in the experiment. For our example, it would be the amount of light present. So that could either be like the time it's in light, the type of light, intensity of light. For our example of light affecting plants, it's how much light is present. And then your dependent variable, or what you measure, is what is being measured. So it can be called both the dependent or the measured variable, and it's really what you're finding. So in our example, it would be the plant height, how much the plant grows. And now finally, one of the most important things in these experiments is having good controlled variables. So control variables are variables kept constant that could change the dependent variable. So they're variables that are kept constant. And for our example, these are all things that could affect plant height. So an example of this would be soil type, the amount of water they're given, the temperature or the surrounding temperature. These are all what stay the same, because if these aren't the same, then you don't really know if it's the independent variable affecting the dependent variable. So now we're going to look at the difference between control and experimental groups. Your control group, or I'll just write C here to make it shorter, is what does not receive the independent variable. And I'll just write IV in second to make it shorter. This is the group that's really at normal temperatures. So for our plant example, we just put the plant in normal light conditions and see how much it grows. So you don't manipulate anything about the independent variable. Whereas the experimental group, or I'll just write E, receives a change in the independent variable. So for this one, it would receive a change in the amount of light. So you could either like stick it in a dark closet with not a lot of light, or you could put it under a lamp that's on like 24 hours. I don't know. Something like that changes the independent variable. And this is how you'll measure the change. So you can compare your experimental groups against your control group to see how it changed from the original. And the final thing you want to know about this are the steps to obtain valid results. So once you've done your experiment and you want to see if you can make it more valid or repeat it, the first thing you want to do is, 1, to repeat the experiment. Your experiments that are designed should be repeatable and that scientists can do over and over again. Conducting the experiment multiple times will allow you to have more data points and to make sure that your data is actually accurate. And the second one would be to increase the sample size. So what this means for our plant example is if we tested 5 plants, we might maybe want to test 15 plants or something like that. So you increase the number of tests, you increase the number of trials, you increase the number of plants in each trial, something to increase the sample size, which decreases the risk of outliers or data that doesn't fit within the data set caused by some random error. Okay, now the last thing in this lesson is graphing. And graphing is very important because most Regents have a graphing question on short response, and people often mess up mostly on this first one. So when the instructions say to, quote, mark an appropriate scale without breaks, the most important thing you want to do is be consistent. You need to have a consistent scale and spacing. And now what this means is that if you're going up on your let's say you're going on your x-axis and you go 2, 4, 6, 8, you don't suddenly want to go 11. You want to make sure that your numbering scale is consistent and that it also goes up per space. So you don't want it to go, one box is 2, the next box is 2, and then you take 3 boxes to do the next 2. You want to make sure it's consistent numbering and consistent spacing. And then the second one, to make sure it's an appropriate scale, is that the graph takes up over half of the grid. So the graph needs to take up over half of the space because you can still have a scale that includes the entire graph, but if the entire graph is only between 2 or 3 points on the scale instead of the 2 or 3 points or grids on the scale instead of the whole thing, then you don't have an accurate or you don't have an appropriate scale, and you would lose points for that. So the next thing they might ask you to do would be to label the x or y axis or title the graph. And the key here is to use the wording already provided in the data table. So if the data table, if you have to label the x-axis and the independent variable given in the data table is amount of sunlight, the time in sunlight in seconds, then your label for the x-axis would be time in sunlight in seconds. So you want to really just use the wording that's always there. And then the other thing you want to remember is the independent variable, or IV, is on the x-axis, and the dependent variable, or dv, is going to be on the y-axis. That's just a thing. If you need to plot it, you just always want to remember that. That will usually be given to you, but that's really just how most graphs are. And the final thing it will tell you to do is to plot the data on the grid and connect the points and surround each point with a small circle. So to do this, you want to make sure you plot carefully. So make sure you're putting your points correctly, and then circle every point. This is just something they ask you to do. You want to put a little circle around each point to make sure you get everything right, and then connect the points with a line. The key thing about connecting the points with a line is that you don't extend past the data points. So sometimes people will always connect back to the origin or 0, 0, even if it's not in the data set, and that would be incorrect. You only want to connect to the points that are in the data set. Okay, so now we're going to go down to do some review questions. And this first question we have is actually a really good graphing question. So we have question 44, 5 groups of corn seeds, each containing 275 seeds, were soaked for 1 hour in different concentrations of gibberellic acid, a plant growth hormone. After 1 hour, the seeds were rinsed in tap water and drained of all excess water. The seeds were then placed on paper towels and kept moist for 7 days. After 7 days, the growing stems were cut and weighed to determine the increases in growth. Then, the percent increases in growth compared to the growth of a group of untreated seeds was calculated. The results were recorded and are shown in the data table below. And here we have our data table. So questions 44 and 45 say to mark an appropriate scale without any breaks on the labeled axis on the grid, and then to plot the data points from the table on the grid on the next page, and surround each point with a small circle and connect the points. So our very first step is to label our axes, or not label the axes, sorry, is to create our scale. So we'll start with the x-axis, which is our independent variable. we'll look at the independent variable. It is the concentration of gibberellic acid, as that's what's being changed. And then what's being measured is the increase in growth. So first we'll look at our independent variable, and we see it ranges from a low of 225 to a high of 600. So we want to do something that ranges from that. So we'll start, let's see, if we can count by 100, it'll go 0, 100, 200, 300, 400, 500, 600, 700. So that works. So for this one, we'll count by 100. So we'll go 0, 100, 200, 300, 400. Okay, so there's our x-axis scale. And now we're going to look at the y-axis scale. So we see that it's increasing growth in percentage, and this ranges from 6 to 15%. So 6 to 30%. So let's see. I think going up by 5 should be work for this. So we'll go 0, 5, 10, 15, 20. So this would be an appropriate scale for both the x and y axis because it successfully fits all the data, and our graph will take over half of the space. So now our next point is to plot the data from the table onto the grid and surround each point with a circle. So first we'll plot carefully. We see 225 goes with 15%, so we'll find 225 somewhere between 200 and 300. That's about a 15% increase, sorry. So that's around there. Next is 330. So we find 30 right there. Then 423 be around there. 515, and finally 606. So there we have our points. Our next step is to surround each point with a small circle. So we're just going to circle every point. And then it says to connect the points. Now on your real test, you'll have a ruler. So your points will be your lines should be straighter than mine. But it should look like that. And now one mistake, and this would be all you have to do for this question, that full points for both 44 and 45. What some people might do, which is incorrect, is to say, oh, we'll just connect it to the origin, so it looks like this. Because 0, 0 is not in your data set, this would be incorrect, and you do not want to connect down to that origin. So now, the final part, there's one other part to this question, which I will show here, which says, identify the control group in this experiment. So if we go back up to the question, it says that the percent growth in growth, and then it says right here, compared to the growth of a group of untreated seeds. That means that because it's being compared to the growth of a group of untreated seeds, our control group would just be that group. So the control group in this experiment is untreated seeds. And that's all we have for this question. Okay, so now we're going to move on to our next question. And this is a short response question, which is really good because it contains both scientific method and lab safety. So it says, poison ivy is a weed that grows in New York State. It synthesizes an oil, urushiol oil, that produces skin rashes. Researchers have found that if poison ivy grows in an environment that contains an increased concentration of carbon dioxide, the plant grows larger, faster, and produces more urushiol. Because carbon dioxide levels in the atmosphere are rising, poison ivy might become a hazard to people who work or vacation outdoors. In order to verify this research, experiments must be carried out. Design an experiment to test whether poison ivy is affected by air containing higher than normal concentrations of carbon dioxide. In your answer, be sure to state the hypothesis the experiment would test, state one way the control group should be treated differently from the experimental group, identify 2 conditions that should be kept the same in both control and experimental groups, describe the type of data to be collected, and identify 1 safety precaution that should be taken during the experiment and explain why it is necessary. So here we have a lot of different points to do, and we'll go through them one by one. So the first one we want to do says state the hypothesis the experiment would test. So we see that in this question it's looking at the effect of increasing concentration of carbon dioxide on how fast the plant grows. So we would say for using this research that we have at the top, we would say if poison ivy grows in increased carbon dioxide, because that's what the question is asking us about, the plant will grow longer, faster, and produce more oil. And this fits our good hypothesis because it says if it grows faster, then it will if there's more carbon dioxide, then it will grow longer, faster, and produce more oil. And this comes directly from the research. So this is fairly just skip the identifying the problem was given to us, and the research is also given to us at the top. So we're starting by creating the hypothesis, which is really the next step in the scientific method. Okay, so our next point is to state one way the control group should be treated differently than the experimental group. So this is really asking what our control group is and why it's different from the experimental groups. So because we're testing how different concentrations of carbon dioxide affect how much the plant grows, our experimental groups will be given different amounts of carbon dioxide. So our control group would really just be regular carbon dioxide. So for our second one, we just say regular carbon dioxide, because the control group is given the normal atmospheric levels, and then that is being used to compare how increasing carbon dioxide will change it. The next one is that identify two conditions that should be kept the same in both the control and experimental groups. This question is asking us really, what are the controls for this experiment? And these are things that would affect the plant growth that's something other than carbon dioxide. So there's some multiple important things like this, like soil concentration, the amount of water, type of poison ivy. So the two on the right are amount of water, because the amount of water will obviously affect plant growth, because if you don't have enough water for the plant, there can't be photosynthesis. And the second one is the type of poison ivy, because we don't know which plant specifically they're testing. They just said poison ivy in general. Different species of poison ivy or different types can all have different growth rates, which means that if we don't have the same type of plant, then our data isn't going to be very accurate. Next is describe the type of data to be collected. So this is really asking us what we're going to be testing. So we can see our hypothesis talks about growing longer, faster, and produce more oil. So we can really test and collect any of those things. For this, in this instance, we can just test how high the plant will grow. So the data we can collect is the height of plants. And then it's always good to just include a unit for how high that plant is. So we'll put centimeters. So the height of the plants in centimeters. Now the final thing is identify one safety precaution that should be taken during the experiment and explain why it's necessary. So one thing we know from the research and about poison ivy is that it produces this oil urushiol, which is bad for our skin. So one safety precaution would be to wear gloves to protect from the oil, because we really don't want to get this toxic oil on our hands. We don't want to be having infections. So we're going to wear gloves to protect from this oil. And that would be everything you need for this question to help get full marks. So that's everything for this lesson. Thanks for listening.